will now be recorded.
So hello everyone, my name is Mariana Pimentel. I'm currently a study engineer at INRAE Bordeaux, and today I'm going to present part of my master work that was submitted to Metabolomics journal, and it was entitled Predictive Metabolomics to Unravel Phenotypic Traits in Pearly Millet Grains from Brazilian Germplasm Panel. This project was coordinated by my supervisor, Mariana Ferreira, my co-supervisor, Milena Barros Santos, and also Pierre Petriac. So to do a brief contextualization, nowadays we are facing the effects of the climate change with the increasing of drought and floods. We also have the stagnation of the main cereals crop like rice and wheat, combining with the ongoing challenge of feeding a population that keeps increasing. All of this intensifies the food insecurity gap and forces us to think about alternative solutions that are more sustainable and resilient. With that, millet emerged as the sixth most produced cereal in the world, most part of the production being in Asia, and 50% of it being of pearl millet. The term millet encompasses several species that produce tiny grains, and here I would like to highlight pearl millet, that in Brazil is mainly used for animal feeding and crop cover, but the grains also hold a great potential for the human nutrition. So as you can see, millet has two main appeals in agronomic and nutritional front. So for the nutritional point of view, it is considered a super cereal. It has high content of lipids and proteins when we compare it to other cereals. It also has some micronutrients like zinc and iron. And it is gluten-free, which makes it a safe solution for people with celiac disease. It also has an interesting composition of phenolic compounds that has different bioactivities, and it has a low glycemic index. In the agronomic front, millet has different morphological traits that make it more resistant to drought and also high temperatures. And because of that, it was very well adapted to the Brazilian Cerrado biome. So when we think about pearl millet and all those nutritional advantages, it's important to address research and efforts for the production of graniferous varieties. And one strategy that we have is to use predictive metabolomics that could bring together metabolomics with the artificial intelligence like machine learning and enable a top-down modeling that use the metabolomic data to understand the mechanisms of the phenotypes. For example, here we have a study that shows the contribution of the plant metabolism to the biotic stress resistance. This technique was also used to predict agronomic traits, for example, yield, and here is our main interest. So with this in mind, our study aimed to predict the phenotypic traits of pearl millet from the grains of the Brazilian active germplasm bank using a predictive metabolomics via machine learning. For this study, we started with a set of 203 genotypes that represented the core collection of the active germplasm bank. And in addition, we also have the information of 21 phenotypic traits. So the grains, they were grounded and extracted with ethanolic solution. It was injected in an Orbitrap system, and then the data was processed with MS-DIO. And after data cleaning and processing, the created features were used to predict the phenotypic traits. And for the prediction, we used two models. We use a random forest on linear model and also generalized linear model with some penalizations. Our data set was split 50% for training and 50% for test. And after that, the features that stood out as predictive markers were identified using an internal standard library with around 450 compounds, and also complemented with a pre-curated plant database from FragHub. So regarding the phenotypic traits, we had 13 quantitative, for example, plant high and also leaf-readed. and 8 qualitatives, for example, grain color and also paniculum shape that is represented here on the right. And these traits, they were selected due to the importance of it for the grain yield, for example, the weight of 1,000 grains. Also, it was important for biomass production, for the response to stress, or because it contributed for the overall plant architecture. Now looking at our results, here we have the overview of the metabolite profile from these 203 genotypes. For constructing this PCA, we used nearly 3,000 features, and these represented, showed the variance of only 36% of the variance in between our genotypes. And most part of it was because of the first principal component. And when we correlate the PCA grouping with the phenotypic, we can see that it was the clusters was mainly different in plant height, number of productive tillers, and also total number of tillers. In general, our PCA was not the best method to explore the genotypic difference in our data set, a lot because of the large number of samples that we had and also the high variability. So because of that, we decided to move forward using supervised analysis to better explore and also model our data. So here we have the box plot showing the predictive performance for both models. So GLM in purple and random forest in turquoise across qualitative on the left and quantitative traits on the right. And for the qualitative traits, we can see that we had a very good performance, and the GLM performed better for 4 traits. And for the quantitative, it wasn't that good, but when we think about a prediction of around 0.5 for traits like 1,000 grains weight, it's already very useful and very meaningful. So here, the random forest outperformed the GLM for 11 quantitative traits. And this difference that we observe between performance and qualitative and quantitative traits can be explained because the quantitative traits, they are usually more polygenic and more influenced by the environmental condition, while the qualitative traits, they tend to be more monogenic and easier to predict. Also, although the random forest were better for most part of the quantitative traits, we decided to proceed with the GLM because it was more suitable for the feature selection. So here we continued only with 15 traits that performed better than 0.5. And here we selected the top predictor features to minimize and avoid the false positives. And then we rerun the model with these refined set features. And here we have the results. So in gray, we can see the performance for using all traits. And in purple, we can see the performance using only the top predictors. And for both qualitative and quantitative, we had really a very good improvement of our model using only the top predictors. Also, when we compared our model to one that was not trained, a random one, we had always a very, very better performance, which is good. And another thing that indicates the robustness of our model was that when you compare, we observed the consistency between the training and the test set. So in theory, this could suggest not much risk of overfitting. So in the end, we were able to improve our model using these predictors. Now here, we can see the Venn diagrams that shows the categories between the qualitative traits. And as you can see, the categories, for example, for flowering range, we had short, long, and discontinuous. And here we don't see much overlap between the categories, showing that the metabolome, it was able to distinct and capture the profiles of each category. Based on that, we decided to proceed only with the unique features for each category. And in total, we had around combining the quantitative and qualitative, we had around 900, almost 900 predictors. Those predictors, they were mainly, we had 49 as level 1 of identification, 68 level 2 of annotation, sorry, and as level 3 we had around 500 almost. And in terms of origin, 63% was from specialized metabolism, while 37% was from the central metabolisms. And to better to simplify actually the interpretation of our data, we decided to group and express the predictors in terms of class and subclass. So here in the sunburst chart, we can see that the classes most representative were actually phenolic compounds, amino acids, fatty acids, and carbohydrates. And among some of those features stood out, some of those metabolites. And I would like to highlight, for example, for plant height, we had N-acetylproline that, like proline, it also participates in the accumulation and regulation during draw stress. For skin thickness, we had the glutamate that is known for the protein biosynthesis and also for the modulation of growth and stress response. And for grain color, we had different phenolic compounds that are normally concentrated in pericarp and in the germ of the grain. And also it has antioxidant activity. So what can we take as the take-home message from here? First, yes, the metabolomics can predict agronomic traits. In our case, we were able to predict 15 from the 21 traits that we're trying, so it's not that bad. The metabolomic profile, it was very specific. And for example, for the qualitative traits, we saw almost no overlap between the categories. And also, although our analysis is very exploratory and it's a study in a single environment, it still provided very useful insights. As future perspective, multi-environment trials and pathway level investigation will be essential for showing the stability and also the biological meaning of our results. So I would like to thank the institution that enabled me to make this work, also my supervisors and teammates, and the RFMF for inviting me to present today. Thank you for your time and attention.
Merci Mariana. On a le temps pour une ou deux questions. Pas de questions. Bon, j'en ai une, Mariana. Finalement, est-ce que c'est utile de s'embêter à faire du random forest si les modèles linéaires performent toujours quasiment aussi bien, voire mieux? Qu'est-ce que tu en penses?
Pardon, je ne pouvais pas comprendre.
Est-ce que c'est utile de s'embêter à faire du random forest qui sont plus
Est-ce que c'est de...
Tu m'entends?
Oui, the first part of, pardon, la première partie de la question, je comprends pas.
What do you think about random forest versus linear models?
Okay, it depends what you're looking for. It depends of how you are doing your approach. In our case, we were starting with on-target method, and our approach was to first select the predictors and then go into the annotation for the identification. Normally, the random forest, due to the way it's organized, the Réseau, the networks, you end up with not a great number of features. You end up with 1, 2, 3 features sometimes. And if you started for only with features that were, for example, already annotated, this wouldn't be bad actually. But in our case, since we started with on target, having only 2 or 3 features is very hard to annotate and identify. So only comparing this situation, for us, it was better to use the GLM. We had more features to annotate after.
Ok, merci.
Thank you.
On te remercie encore, Mariana, et on passe à Mazzarine.
Okay, can you hear me? Okay, all my apologies in advance because I'm in the middle of somewhere with a lot of people, so I hope you can not hear them. I want to. Can I do that? Okay, what do you see now?
We see the screen and little windows that you can close, and then it's fine.
Okay, perfect. So I can. Now you must have just the presentation. That's it?
Yes, perfect.
Okay, perfect. So, hello everyone, thanks to be here for this presentation. I'm Mazzarine Laboureau. I'm a PhD student in chemical ecology at the University of Neuchatel. I'm going to present you one of my projects, the hidden impact of sampling method on plant metabolomics data. So, to give you more general context, ecological and metabolomic studies span a broad continuum, ranging from highly controlled laboratory experiments on one hand, to in natural studies conducted in remote environments on the others. On one side, we have standardized, easily accessible data generated under controlled conditions. And on the other, we obtain unique and irreplaceable data set with high ecological value because they're directly associated with biotic and abiotic factor of the natural environment. But regardless of where we position ourselves along this continuum, we face a major methodological challenge when comparing this data, because to compare field data with laboratory standards, enzymatic activities must be rapidly halted after harvesting. For that, generally, we use liquid nitrogen as a gold standard because it stops directly a metabolic reaction, but in other way is bulky and hazardous and subject to a rapid evaporation, and that's impracticable to extend in field campaign. There is other method that we can use is, for example, dry ice, feed extract, or different drying methods. But we have a trouble with this method because the dry or freeze, this slow dry or freeze, oh, I'm gonna come back, sorry. They dry or freeze plant tissue more slowly. And this slow process can induce stress response in the plant. And the plant gonna produce some specialized compound, defense compound, which in turn alters the metabolomic profile. So the main question here is to understand, to study, therefore, the assess and quantify the impact of this preservation method on plant chemistry. For that, to address this question, we selected 10 plant species from the Neuchatel Botanical Garden, providing a broad representation across the plant phylogenetic tree here. And we tested 6 different methods. So the first one is liquid nitrogen. So samples were rapidly frozen under 180 degrees in liquid nitrogen. And that's our reference method for the rest of the experiment. We have also the dry ice. So samples were rapidly frozen by placing them in contact with ice at minus 80 degrees. For the feed extract method, we obtain a small leaf cutout mixed with 1 milliliter of solvent mixture. Our solvent mixture is a mix between methanol, water, and formic acid. We have also a oven drying method, so samples were dried in oven at 40 degrees during 72 hours. For the silica gel, we just put the leaf in coffee filter in a waterproof bag full of silica gel during 10 days. And for the air drying, sample was stored in a coffee filter and let in the air during 10 days. So for that, we obtained 5 samples per plant for each method, so a total of 300 samples. And now for the results. Here you can see a chemical dendrogram. So each tip label represents a sample, and they are ordered according to their chemical similarity. Colors indicate species, and the tip label also specifies the sampling method used. So here we can observe a good clustering for several species, for example, Laburnum anagyroide, Persa americana, or Pteridium aquilinum. But in the other hand, 4 other species, Brachychaeton populineus, Strelitzia reginae, Celastrus candens, et Ruellia squarasa, show a less stable clustering, particularly for the field extraction method. And we can also see here a T-SNE, so each sample is positioned according to its similarity to its 40 nearest neighbors, and the analysis was repeated 1,000 times. The fact here that samples are not clustered in the center indicates that there is a clear species effect here. And we can see a difference in the ellipse size reflects species-specific response to the treatments. Species such as Laburnum anagyroides, Pteridium, et Persa americana. So these species show a high stability with small ellipse, whereas in the other end, Ruellia, Centaurea, Strelitzia, or Brachychaeton display a larger ellipses indicating a greater variability. Overall, the T-SNE analysis confirms the observation from the dendrogram distinguish stable species from more variable ones. Here you have a heat map comparing the mean peak intensities for each superclasses across treatment relative to the liquid nitrogen reference. So we can see, for example, here over detection of fatty acid in the drying method. And if we look more in detail, the effect is particularly pronounced for glycerophospholipid, octanodecanoid, or glycerolipid. And we can see also something interesting here, another expression of sesquiterpenoid in the feed extraction, but an under detection of this compound in over drying methods. And from statistical perspective, metabolite superclass have a significant effect on peak intensities, while treatment also show a significant effect, but with smaller magnitude, showing that preservation method alter metabolite profile in a way. And the interaction between treatment and superclass are not significant, so there's no effect on that. And the residual variation is relatively low, so that confirms the main source of variation are captured by the treatment and the superclass. And now you are an upset plot. So on the left here, you can see the average chemical diversity for each treatment, and we can observe a clear loss of chemical richness with the feed method extraction. And in this part of the plot, you have the number of compounds shared among groups of 6, 5, 4, 3, 2, or 1 treatments. Something interesting here is this one. So we can see that a large number of compounds are common among our 6 treatments with 2,000 compounds in common. Here, this group are all the group together excluding feed extract 1, and we can see that 3,350 compounds are in common in this treatment. So in other terms, these compounds are not available in the feed extract method. The group of 4 treatments do not reveal any clear informative pattern, but in contrast, for the group of these 3 treatments, so it's the drying one, they shared 250 compounds, notably fatty acid here. And there is, for the group of 2 treatments, the freezing method, so here, with about 200 compounds in common. And finally, for the feed extraction method, show around 250 compounds unique itself, meaning compounds detected only in this method. So overall, this analysis highlights 3 distinct groups of preservation method: drying ones, drying is one, freezing is one, and the feed extraction method. And 2, discuss the results. The divergence observed for Brachycheton, Celastrus, Strelitzia, and Ruellia mainly reflect a high intra-species variability, rather than an effect of the preservation method themselves that could be explained by specific biological traits such as variable accumulation of triterpene and cardenolide, which are strongly influenced by light exposure and mechanical stress. And in addition, some of these species exhibit a highly plastic metabolism, particularly for polyphenol and polysaccharide, which is closely linked to hydration stages. So in conclusion, overall, these results show that the species' specific physiology and evolutionary history play a major role in determining metabolite stability and detectability. And finally, to compare all these methods together, liquid nitrogen is a gold standard because it's instantly stop metabolism reaction, but it's expensive and difficult to use in field condition. Dry ice provides a metabolic profile very close to liquid nitrogen with only minor loss, making a strong practical feed alternative. Feed extraction avoids the cold chain, but causes strong under detection of labile metabolites due to enzymatic activity and oxidation. And drying methods are easy to implement, but induce artificial drought stress response, leading to over-detection of lipid and stress-related compounds. So finally, there is no perfect method. Each involves a trade-off between metabolomic fidelity and field feasibility and cost. Liquid nitrogen remains the best option, but under feed contrast, dry ice or silica gel are viable alternatives, as long as they're associated to analytical bias and taken into account. I would like to thank my supervisor, Emmanuel Defossé and Sergio Rasman, Blaise Muholzer, the director of the Botanical Garden of Neuchatel, and Gaëtan Gloser for his help to the analysis of the data. And thank you for your attention.
Merci beaucoup, Mazzarine. Très intéressant. Est-ce que vous avez des questions? Vous êtes timide aujourd'hui. Quelqu'un lève la main. Cyril?
Oui. Bonjour.
Bonjour.
Vous m'entendez?
Oui.
Très bien.
Parfait. Merci pour cette présentation. J'ai une question méthodologique. En fait, le CO2, le dry ice,
oui.
Est-ce que vous l'avez gardé, comme vous le montrez, en petits bâtonnets? Ou est-ce que vous avez essayé de le sprayer sur l'échantillon, enfin, lors de l'échantillonnage?
Non, on avait des box de.
Ok.
On avait des boîtes avec les glaçons de glace carbonique dedans et on enveloppait les feuilles dans des aluminium foil, des feuilles d'aluminium, et après on posait juste les feuilles en contact avec la dry ice.
Ok. Ouais, parce que clairement, si vous utilisez comment on dit ça en français, un distincteur à CO2,
oui,
vous avez le même effet en fait.
Oui, je m'en doute bien, mais. Mais
et
pareil, je pense que c'est compliqué
ici.
à utiliser sur le terrain. Enfin, trimballer un extracteur, je pense que ça va être difficile.
C'est pour dire que peut-être qu'il y a des biais aussi que vous n'avez peut-être pas tout à fait mesurés dans les différentes méthodologies. Liquid nitrogen, c'est utilisable aussi en spray. On peut le sprayer sur les feuilles. Enfin voilà, il y a des techniques intermédiaires qui existent pour faire du sampling au champ.
Mais si on spray le liquide nitrogène sur la feuille, on n'a pas ce risque d'évaporation et au final, la feuille va décongeler au bout de quelques minutes.
Oui, mais c'est juste pour le en fait, ce qui peut, enfin, en termes expérimentaux, ce qui existe parfois, c'est que pour éviter d'avoir le wound effect, l'effet blessure sur la plante, vous faites votre spray de liquid nitrogen et après vous coupez l'échantillon et vous le plongez directement dans un tank. Donc en fait, c'est entre guillemets une anesthésie locale avec prélèvement rapide et après évidemment vous avez bien sûr le tank d'azote liquide à proximité. Donc là il y a toujours cette problématique là de logistique. Et pour le dry ice c'est pareil, vous pouvez faire un mini extincteur à CO2, vous sprayez sur la plante, vous cuttez et vous plongez dans le dry ice. Et là, vous limitez en fait, enfin moi en tout cas c'est mon point de vue, hein, les gens qui utilisent ça, je pense qu'ils limitent en fait le timing entre le wound, le prélèvement sur la plante, vous limitez la dégradation métabolique, enfin l'évolution métabolique, pardon, entre le cutting et la congélation.
Bah, on se retrouve de nouveau face au trade-off entre quelque chose de vraiment proche de la réalité, mais difficile à utiliser sur le terrain, parce qu'il faut donc la dry ice ou le tank de liquide nitrogène et l'extincteur, ou avoir quelque chose de plus facile à utiliser, mais probablement de moins précis. C'est tout un trade-off. Ça dépend ce qu'on voulait faire, quoi.
Ok. Merci. S'il n'y a pas d'autres questions, j'en ai une deuxième, mais je vais laisser peut-être d'autres personnes.
On a le temps pour une deuxième question? Oui. Vas-y, Cyril.
Ok. Merci. Est-ce que vous avez pris en compte la communauté microbienne épiphytique?
Pas du tout.
Ok.
Pourquoi?
Parce que là aussi, vous avez des problématiques. Vous avez des producteurs de métabolites qui sont à la surface de la plante et qui peuvent eux aussi réagir peut-être de manière différente à vos méthodologies.
Ok.
Voilà. Merci. En tout cas, très belle présentation. Merci beaucoup.
Merci.
Merci beaucoup, Mazzarine. Et on enchaîne avec Eurydice, si elle est prête.
Normalement, Eurydice, tu as les droits maintenant, donc tu peux présenter.
Ouais. Hop. Est-ce que vous y voyez quelque chose ou pas?
Oui, on voit ton mode présentateur et pas ton mode plein écran.
Ouais, c'est ça le problème. Alors, non. Ok, attends. Désolée. Est-ce que là, vous voyez quelque chose?
Ah, c'est bon.
Super, bon, on va faire comme ça alors. Hello everyone. So I'm Eurydice, and first I would like to thank Theo and Pierre to invite me to present you my work today. So I'm a third year PhD student at the Biological Station of Roscoff with Catherine Leblanc, and at the National Museum of Natural History in Paris with Swazik Prado. And I'm working on the vanadium-dependent alloperoxidase rules in brown algae, so mostly Ectocarpus sp7, using knockout mutants and metabolomics. So first, why brown algae? As we can see on this tree of life, brown algae evolve very independently, and they are distant from fungi and animals, but also from green lineages to which belong green and red algae. So this independent evolution makes them very interesting and to study because they present original traits as defense mechanisms involving halogens. Indeed, as we can see on this graph, which represents the total iodine and bromine content in some brown, but also green and red macroalgae, we can see that brown macroalgae accumulate a lot of iodine and also bromine. And for instance, in Laminaria digitata, the total content of iodine is up to 1% of its dry weight. And in this macroalgae, the bromine and iodine are mostly present in the peripheral tissues. But we don't know what mechanisms are involved in the uptake and the release of these halogens, and what are the biological roles of these halogens in algae. An enzyme called vanadium-dependent alloperoxidase is likely to be a key enzyme in halogen metabolism. Indeed, in presence of hydrogen peroxide, it can oxidize halides in order to form a reactive intermediate, XOH, which can react with the organic substrate in order to form organohalogens, or react again with the hydrogen peroxide in order to detoxify the cells. Other hypotheses have been proposed in order to explain the different roles of this enzyme, vanadium-dependent alloperoxidase. So they could be involved in the uptake and the chemical complexation of halides, but also in the resistance to oxidative stress and in the defense and signaling mechanisms during biotic or abiotic interactions. That's why the main aim of my thesis is to understand the roles of the VHPO and the halogen metabolism, and also the associated mechanisms through functional genomics and halogen metabolism studies in Ectocarpus sp7. So Ectocarpus sp7 is this little macroalgae, brown macroalgae, and one algal type VHPO gene exists in this species, and it's a code for vanadium-dependent bromoperoxidase, so it can oxidize the bromide and the iodide. And at the biological station of Roscoff, 3 CRISPR-Cas9 knockout mutants have been generated. And now it's important to note that this knockout, this BPO mutation, requires the knockout, so the mutation of the APT gene for the selection. So we have 3 types of strains: the wild type, the APT, which is a knockout for the APT gene, and the BPOs, which are a knockout for the APT and also the BPO gene. So here you have the structural modeling of the vanadium-dependent bromoperoxidase. So in green, it's the wild-type bromoperoxidase. And in the other colors, it's BPO1, BPO2, and BPO3, the 3 mutants generated. So these 3 enzymes, BPO1, BPO2, and BPO3, are not functionally active. And in this presentation, I will present you the impact of this VBPO knockout on the metabolic patterns for metabolomics characterization of each strains, and also the impact of the application of an oxidative stress. So first, the characterization of each strains through metabolomics. So I carry out a classical extraction from the alga in powder. So for the 7 strains, wild type, 3 APTs, and the 3 BPOs. So 2 extractions, one with methanol and the other one with etat acetyl. And I run this extract in LC-MS-MS with C18 colon in positive and negative ionization modes. And the data were analyzed following this workflow with MZ9-4, and the statistical analysis is performed with MetaboAnalyst, and I built a molecular network with GNPS2, and I tried to annotate some VAPs with the spectra and some databases. So first, here you have the 4 PLSDA obtained. So at the top, you have the positive ionization mode, and at the bottom, you have the negative ionization mode. On the left, the etat acetyl extraction, and on the right, the extraction with methanol. And we can say that the BPO strains are always between the wild type, so in green, and the APT on the left of the PLSDA on the component 1. But BPO2 and BPO3, so in yellow and in blue, appear to group together. So we can say that each type of mutation causes a specific change in the metabolic pattern, but we have also a strong effect of the APT mutation. However, some VAPs can be highlighted and they are underexpressed in the BPO strains. And these VAPs belong to the beta-in lipids family. Beta-in lipids are non-photosynthetic membranes. So we were currently wondering what is the link between these beta-in lipids and the enzyme vanadium-dependent bromoperoxidase. However, some beta-in lipids are present in certain BPO strains. So my hypothesis is that we have only one type of the beta-in lipids which are underexpressed in the BPO strains. And to answer, I'm currently analyzing the run with a lithium adduct in order to distinguish the two forms of the beta-in lipids, which are two isomers. So next, we will see the impact of the application of an oxidative stress. So first, on the physiology of the algae. So first, I selected 3 strains, one wild type, APT4 and BPO3. APT4 and BPO3 have the same APT mutation, and I apply 0.75 millimolar of hydrogen peroxide on the algae, or H2O2 for the control, and I measure the photosynthetic yield with PAM. And on the results, we can see here it's photosynthetic yield as function on the time, and we can see that the treatment with the 0.75 millimolar of H2O2 significantly reduces the photosynthetic capacity of the strains. And within the first 8 hours, after the application, but wild type in green recovers its photosynthetic capacity more quickly than BPO3 strain in orange. So we can say that the VBPO, the enzyme, appears to be involved in regulating this oxidative stress. And now for the metabolomic characterization, I performed the mostly same protocol with wild type APT4 and BPO3 in quadruplicates, and I apply also 0.75 millimolar of hydrogen peroxide. And I harvested the algae and carrying the PAM measurements 24 hours before the treatment, 4 hours, 8 hours, and 24 hours after the treatment. The harvested algae is for the transcriptomic analysis performed by Serena and Olivier. And I also carrying the metabolic extraction for the metabolomics analysis. So it's the same protocol as before, but only with methanol. And now it's mainly lyso-ornithine lipid, which are found as underexpressed in BPO strain. However, these lipids are characterized as bacterial membrane lipids. So we have to do metabarcoding on another method in order to study the microbiota of the algae on different strains. Also, lyso-ornithine lipids highlight its VIP for all the time and all the treatments, so unstressed and stressed strain. So we searching into stress response molecules, and all these results will be put in parallel with transcriptomic analysis. To conclude, we have seen that each strain presents its own metabolic profile, but some metabolites as beta-in lipids are less abundant in KO-BPO strains. And to go further in these results, we are performing lipidomic analysis in REN. And also we have seen that 0.75 millimolar of hydrogen peroxide reduces the photosynthetic yield for up to 8 hours in the BPO strain, and it should finally disrupt the metabolome. So ongoing research is performing to decipher the VBPO mediating regulations involved in oxidative stress response. And the study of metabolic pathway involved in the resistance to oxidative stress will be carried by transcriptomic analysis. And the last but not least, we are also searching volatile, maybe halogenated compounds involved in the VBPO pathway and also in the response to the oxidative stress. And we carrying that with SBSC at REN. And with that, I would like to thank my supervisor and all the people involved in this project and you for your attention. Thank you.
Merci bien. Des questions? Alors moi, j'en ai quelques-unes. Comment tu comptes en fait collecter tes volatiles? Puisque l'algue, elle est dans l'eau, c'est dans la solution, on va dire, marine.
Ouais, alors on a fait plusieurs tests pour tout te dire qui n'ont pas marché parce que du coup on a fait des extraits d'algues de base, mais sauf que tous les volatiles s'étaient envolés. Et aussi on a fait... En fait là ce qui a marché c'est que j'ai fait de la SBSE, du coup à P2M2 à Rennes avec Léo. Et en fait du coup on met des barreaux dans le milieu. Ce qui a marché, en fait, c'est quand on mettait 400 mg d'heure à 72 heures d'incubation et tu mets ton barreau qui a une matrice de PDMS et du coup qui absorbe les composés qui sont émis dans l'eau. Et ensuite, tu les désorbes avant d'être injecté dans l'AGC. Et ça nous aurait peut-être donné des bromophénols.
Ok. Et tu n'as pas de précurseur halogéné en LCMS?
Je n'ai pas trouvé, non.
Ok. Et mon autre question, c'est tes mutants knockout, est-ce qu'ils ont un phénotype déjà? Est-ce qu'ils sont affectés en termes de croissance ou autre?
Bah ça non, j'ai fait des tests justement de croissance et non, il n'y a aucun... Quand tu les stresses fort, ils répondent beaucoup plus fortement, même visuellement, ils deviennent blancs, ils meurent quoi.
Oui.
Mais autrement, ils grandissent, c'est APT plus qui a l'air de pousser plus vite et c'est plus lui en fait à chaque fois qui sort vraiment du lot. Finalement, la double mutation semble plus s'apparenter au wild type que la simple mutation APT.
Ok, merci. Est-ce que vous avez des questions?
Oui, Cyril?
Oui, rebonjour. Merci pour votre présentation. Moi, je suis surpris qu'il n'y ait pas de... Je ne trouvais pas d'effet sur le métabolisme central. Est-ce que c'est lié aux conditions au workflow de métabo que vous mettez en place, type de solvant, etc., ou est-ce qu'il y a quelque chose que j'ai loupé?
Alors, métabolisme central, c'est-à-dire?
Ouais, des sucres, des acides organiques.
Ok, bah après ça, j'ai trouvé du coup ces lipides-là qui ressortaient vraiment parce qu'ils étaient vraiment sous-régulés, enfin limite absents même de chez mes BPO. J'ai pas fini du tout la notation. Après les sucres, du coup, il y a une autre personne au labo qui est en train de les regarder. Elle m'a pas donné les résultats encore.
Ok. Parce que l'oxygène, le peroxyde d'hydrogène, bon, oxygène peroxide,
Ouais.
hydrogen peroxide, pardon, il y a de multiples effets sur un organisme.
Oui. Après, c'est pas une dose non plus extrême.
Oui, c'est vrai.
On voulait juste en fait induire un léger stress sans complètement tout déréguler, quoi.
Ok, ouais, peut-être c'est trop rapide, trop subtil pour avoir des effets plus délétères sur le métabolome. C'est ça que vous voulez dire?
Ouais, peut-être. Et en tout cas, quand j'ai fait ma PLSDA, du coup, c'était pas tout à fait correct parce que j'ai pas aligné tout le monde en même temps, mais ce qui semblait se passer, c'est que il y avait plus un effet de temps qu'un effet de traitement, en fait.
Okay.
Voilà.
Merci.
Merci à toi.
Merci. On va clôturer la session alors. Et on vous souhaite de belles fêtes.
Merci à vous aussi.
Merci.
Au revoir.